Good morning. Welcome to today's class. This morning we want to discuss Krebs cycle. This is also known as Tricarboxylic acid cycle or citric acid cycle. In the video before this, we look into glycolysis. We will realize that in glycolytic pathway, a molecule of glucose will be broken down with the help of cofactors, coenzymes and enzymes to their last part, which is 2 molecules of pyruvate. And we summarize it this way. From glycolysis, the 1 molecule of glucose will be broken down to pyruvate, 2 molecules of pyruvate. So 1 molecule of glucose will finally be broken down to 2 molecules of pyruvate. And we sketch these 2 molecules of pyruvate this way. We sketch it this way. So we say this is 2 molecules of pyruvate. So it has methane, that is CH2. It has carbon with bonded by oxygen. Then you also have the COO, that is pyruvic acid, that is carboxylic acid. So we realize that this glycolysis, after breaking down 1 molecule of glucose, it will give you 2 molecules of pyruvate with other products we have. But we want to discuss these 2 molecules of pyruvate first. So these 2 molecules of pyruvate will also undergo decarboxylation reaction, that is oxidative decarboxylation. So once it undergoes this, it will use COH and produce CO2. As it produces CO2, this will be used in the reaction to produce this. We call this nicotinamide adenine dinucleotide, and we call this reduced nicotinamide adenine dinucleotide, because we say that this pyruvate here is 2 molecules. It means that here will be 2 molecules, here will be 2 molecules, everything here will be 2 molecules. Now, this will take place by a process called oxidative decarboxylation. And the enzyme involved is called pyruvate dehydrogenase. Pyruvate dehydrogenase. By an enzyme, pyruvate dehydrogenase. So when this pyruvate dehydrogenase breaks down 2 molecules of pyruvate, it will be giving us what we call acetyl-CoA. Acetyl-CoA, acetyl-CoA, acetyl-CoA. Then we have a double bond here. So we have CH2, we have another carbon with double bond oxygen, we have sulfur, and we have CoA. This is called acetyl-CoA. Now, this acetyl-CoA can also be gotten from beta oxidation of fatty acids. That tells us that when fatty acids is oxidized by beta process, you have acetyl-CoA as well. Then you can also have acetyl-CoA by breakdown of 2 pyruvates to get it. So this will also be 2 molecules. 2 molecules of acetyl-CoA. Now, from this acetyl-CoA, it will enter the tricarboxylic acid cycle or Krebs cycle or citric acid cycle. From here, it will enter this cycle we talk about. Here, it will also give you a reaction will take place here, and the enzyme involved here is called citrate synthase. Citrate synthase. This enzyme will act on acetyl-CoA. Once it acts on acetyl-CoA, there will be production of CoAs. Once this is produced with the presence of, and there is a reaction called condensation. There will be condensation reaction here. That is, water will be needed, or there will be production of water in this reaction. So once this condensation reaction takes place in the presence of citrate synthase and they act on this acetyl-CoA, what you will be having is citrate. So this will produce what we call citrate. CH2COO, C, then COO. This is citrate. This is citrate. We put COO and hydrogen here. Citrate. So still two molecules of citrate. So this is what we call citrate, the chemical structure of citrate. Then this citrate will also undergo dehydration reaction, and once you undergo dehydration reaction in the presence of aconites, it will produce isocitrate. Isocitrate. Aconites enzyme, then dehydration reaction will take place. So this aconites is the enzyme responsible for breaking down citrate to isocitrate. So what is the structure of isocitrate? You have it to be we put CH2 here, then we also put this is hydroxyl. So this is isocitrate. That is a compound that will be formed when this citrate is broken down by aconites in the presence of dehydration to give you this. Then this will be two molecules. Why? Because the pyruvate we start from is two molecules. Now what do we do next? This isocitrate will undergo oxidative decarboxylation. Once it undergoes oxidative decarboxylation in the presence of dehydrogenase, isocitrate dehydrogenase, it will act in the presence of isocitrate dehydrogenase. Then in this, there will be enzyme or coenzyme that will act here. 2NAD plus will be giving us 2NADH plus hydrogen ion. So this will be produced. Then isocitrate, after acting by isocitrate dehydrogenase, it will be giving us alpha ketoglutarate. alpha ketoglutarate. Then that is 2 molecules of it. Also 2 molecules. Then now is what is the structure of alpha ketoglutarate? We can sketch it this way. We can sketch it this way, that this is, we now say CH2 double bond, then we say C, this is carbon, this is alpha ketoglutarate. That is the structure of it. Then after having it, this alpha ketoglutarate will also undergo another reaction with the help of alpha ketoglutarate dehydrogenase. So this will act by alpha ketoglutarate dehydrogenase. So this is the enzyme that will catalyze this. This is the enzyme that will catalyze alpha ketoglutarate. Once it catalyzes it, it will produce CO2. So there will be oxidative decarboxylation here. There will be oxidative decarboxylation. Then another thing that will take place here is that this NAD plus will produce 2 NADH plus. So, and there will also be production of CO2. Okay? So, once this enzyme acts on alpha glutarate, we'll be having what we call succinyl-CoA. Succinyl-CoA, 2 molecules of succinyl-CoA. 2 molecules of succinyl-CoA. Now, this succinyl-CoA have this structure. It has this structure to be this way. So you put double bond oxygen. This is called 2-succinyl-CoA. We have to because from the start we have 2 pyruvate, so they will move like this to get 2-succinyl-CoA. Then when we get to 2-succinyl-CoA, another step will take place by an enzyme called succinyl-CoA synthetase. Succinyl-CoA synthetase will act. Succinyl-CoA synthetase will act by the process of phosphorylation. There will also be phosphorylation here. Then another thing that will happen here is that this glutamate diphosphate, GDP, 2 molecules of it will go into reaction to produce 2-GTP. And this 2-GTP can also be called 2-ATP. 2-glutamate triphosphate is something as 2-adenine triphosphate. So as it gives us this, by this enzyme called succinyl-CoA synthetase, what we have is that as this acts on 2-succinyl-CoA, it will be producing succinate. 2 molecules of, so this will give us 2 molecules of succinate. Now, what is the structure of succinate? We have CH2, CH2, then we have, so this is the structure of succinate. So there are 2 because all these are in 2s. This one is in 2. Okay? Then this succinate will be acted by another enzyme called succinate dehydrogenase. Succinate dehydrogenase. As this succinate dehydrogenase acts on this succinate, there will be need that the coenzyme FAD will go into the reaction to produce FADH2, reduce flavine adenine dinucleotide. So as it gives us this succinate dehydrogenase, as it acts on this, what it will be giving us is fumarate. 2 molecules of fumarate. Okay? As we have 2 molecules of fumarate, that 2 molecules of fumarate has these structures. This is 2 molecules of fumarate. 2 molecules of fumarate. Okay? So this is a typical structure of 2 molecules of fumarate. Then the fumarase will also act, fumarase enzyme will also act in the presence of what? Hydration. Hydration reaction, that is the presence of water. It will act to give us what we call malate. So malate has this structure to be this way, that so 2 molecules of it is what it will give us. 2 molecules of fumarate. So this is typical structure for 2 molecules of fumarate. Then from here, let's put it well. From here, we say fumarase will give us this structure. This is called malate, 2 molecules of malate. Now, this malate we have will also undergo another reaction to give us, in the presence of malate dehydrogenase, will undergo another reaction in the presence of the catalyst malate dehydrogenase, and in the presence of this coenzyme to produce these coenzymes. Okay? So once it undergo reaction, it will be giving us oxaloacetate, 2 molecules of oxaloacetate. And what is the structure of 2 molecules of oxaloacetate? We have it this way. we have it to be two molecules of oxaloacetate. So this is structure of two molecules of oxaloacetate. So it is this two molecules of oxaloacetate that will also undergo, that continue with this. So it will follow from two molecules of oxaloacetate and move to give us this. So the process continues that way. It continues that way. So what we realize here is that this is a typical structure of what we call citric acid cycle. We call this two molecules of oxaloacetate. From here, we call this one citrate. Citrate. Okay? Then from here, it gives us this. Now, what is the essence of this pathway? In this pathway, we are able to see that 2 ATP were produced. 2 ATP was produced because we said that 2 GTP is also taken to be 2 ATP. And we also notice that the number of reduced nicotinamide adenine dinucleotides we have are this one, 2 ATP and 2 molecules. From pyruvate, we have 2 molecules here. Another molecule is, we have another 2 molecules here, 4 molecules of this. We have another one, 6 molecules. Then we have this is 6 molecules. Then we have another one here, 8 molecules. Then we have 8 molecules of NADH from TCA, from Krebs cycle or citric acid cycle. Now remember, in glycolysis we have extra 2 molecules of this there. We have extra 2 molecules. This is from glycolysis and this is from glycolysis and this is from TCA cycle. Whereas this is for TCA. In glycolysis, we also have 2 molecules of ATP produced. So this one we have here is from TCA, while this one we have here is from glycolysis. Then also from TCA, we have 2 molecules of FADH2. 2 molecules of FADH2, that is the reduced flavine adenine dinucleotide. So we have seen the total NADH we have. NADH we have from glycolysis and TCA are 8 plus 2, 10 NADH. Then we also have 2 FADH. Then in the next video, we will see how to convert all this we have. Okay? See the next video so that we see how we realize the total ATP produced by each breakdown of glucose in the body. And as the cell uses it, how many ATP, energy form of ATP is the cell making use of. We will see how the conversion is being done. See the next video so that you see how we do it. Thanks for watching the video. Please don't forget to click like button or subscribe as you watch it. See you in the next video.